Hello, this is Bob Rutledge. In this series of presentations, I will describe how LRE was developed and how it can be used to conduct absolute quantification. I will start by pointing out that our 2008 article in BMC Biotechnology provides a detailed introduction to LRE, along with additional material that will not be covered in this overview. Our second article, published in BMC Molecular Biology, extends this work by providing an in-depth investigation into methods used to determine amplification efficiency. Our third article, published in PLOS One, investigates the performance capabilities of LRE, along with additional details as to how LRE is implemented. Note that most of the datasets presented in this overview were taken from this last article. The series will start with describing the origins of LRE, along with how target quantity is determined. This will be followed by a description of how quantitative scale can be established using lambda genomic DNA as a universal quantitative standard. In the final presentation, I will describe methods that can be used to verify quantitative accuracy. So what is LRE? LRE is simply a collection of methods that provide a number of capabilities beyond that possible with current methods. Foremost is the ability to conduct absolute quantification without the need to construct target-specific standard curves. LRE also provides extensive quality control capabilities, including the ability to determine the amplification efficiency within individual PCR reactions. Although not addressed in this overview, LRE is also amenable to software automation, providing the ability to develop high-throughput applications for absolute quantitative PCR. Originating from attempts to develop a method for determining amplification efficiency that did not require construction of a standard curve, LRE was derived from analyzing the kinetics of PCR amplification. This was based on defining amplification efficiency as a relative increase in amplicon DNA over a single cycle. In this approach, amplification efficiency for cycle C is determined by the increase in reaction fluorescence, defined here as Fc, relative to the previous cycle, that is Fc minus 1. This is referred to as cycle efficiency, or EC, which is generally expressed as a percentage. To illustrate this method, I will focus on an amplification profile produced with 100 femtograms of lambda genomic DNA, an amplicon called Cal1, and QIAGEN's Quantitect enzyme formulation. Note also that standard curve analysis indicated that amplification efficiency of this assay is close to 100%. This table lists the fluorescence readings within the central region of the profile, which are denoted by the red circles, with the EC values for each respective cycle listed in the last column. The most obvious outcome of this analysis is the prediction that a progressive loss in amplification efficiency occurs across the entire amplification profile. In addition, this loss is not gradual, but rather results in a substantive loss in amplification efficiency, even within the earliest cycles of the profile. The kinetics of this loss can be further illustrated by plotting cycle efficiency against reaction fluorescence. This reveals a linear relationship between the loss in amplification efficiency and amplicon DNA quantity. Although we have not attempted to characterize the underlying mechanism of this linear coupling, this plot provides many insights into the kinetics of PCR amplification, which were essential to the development of LRE. Most evident is that amplification efficiency is defined by the y-intercept. That is, the maximal amplification efficiency, referred to as Emax, which is produced in the absence of amplicon DNA. It is also apparent that the maximal fluorescence, called Fmax, is defined by the x-intercept. Most significant, however, is that the dynamics of PCR amplification can be described using a linear equation where the slope defines the rate of loss in amplification efficiency, referred to here as delta E. This in turn allows values for delta E and Emax to be determined using linear regression analysis. Called linear regression of efficiency, this is where the LRE acronym was derived. For this profile, this produced an R-squared of 0.9994 and an Emax of 100.2%. Note also that Fmax can be determined from the ratio of Emax to delta E. The close correlation of Emax with that predicted from standard curve analysis provides support for the general validity of this approach, at least for amplification efficiency determination. Answering the more general question as to how LRE analysis can be applied to quantitative PCR required returning to earlier work, which involved the application of sigmoidal mathematics to quantitative PCR. As described in a previous study, quantitative PCR can be conducted by modeling PCR amplification as a sigmoidal process using two equations derived from the classic Boltzmann sigmoid function. The first equation allows target quantity, expressed in fluorescence units, to be calculated once values for Fmax, C1/2, and K have been obtained. This can be done using nonlinear regression analysis to fit the fluorescence readings within a profile to the second equation. Referred to as sigmoidal curve fitting, this approach proved to be difficult to implement due primarily to distortions within the plateau phase. Nevertheless, this work did illustrate the ability of sigmoidal mathematics to provide several capabilities not possible with conventional methods. It was therefore of interest to investigate how Fmax, C1/2, and K relate to delta E and Emax. As described in our BMC Biotechnology article, Fmax can be calculated directly from Emax and delta E, whereas K is defined by the inverse ln of the amplification efficiency. Substituting for K in the second equation allows C1/2 to be defined in terms of Emax, Fc, and cycle number. This in turn allows substitution of C1/2 in the first equation, which produces an equation that allows fluorescence readings to be converted directly into target quantity. Similarly, substituting for K in the first equation allows C1/2 to be defined in terms of Emax and F0. This in turn allows substitution of C1/2 in the second equation, which produces an equation that allows reaction fluorescence to be predicted for any cycle number. Let's then have a look at how these derivatives can be used. Returning to the profile presented earlier, linear regression analysis is applied to the cycles within the central region of an amplification profile, providing values for Emax and delta E. Denoted here by the red circles, this contiguous group of cycles is referred to as the LRE window. Using the first LRE derivative, F0 values are calculated from the fluorescence readings from each of the cycles within the LRE window. An average F0 is then calculated along with a coefficient of variation. The 0.6% CV generated here is typical for high-quality profiles and reflects the high level of precision that can be achieved with LRE analysis. The primary output of LRE analysis is therefore an average F0 value that is typically derived from 4 to 8 determinations depending on the size of the LRE window. The analysis can also be presented graphically by plotting the F0 values against reaction fluorescence. Referred to as the F0 plot, this illustrates visually the similarity of F0 values within the LRE window. Importantly, this plot also reveals a rapid divergence in the F0 values derived from cycles close to the plateau phase. Although I will not go into the details, this abrupt divergence can be used to automate selection of the LRE window. Indeed, it is this type of recursive analysis that is central to the robustness of LRE. This then leads to the final aspect of LRE analysis, which is the ability to model PCR amplification. Using Emax and delta E, along with the average F0, reaction fluorescence can be predicted for each cycle within a profile using the second LRE derivative. This table summarizes the results produced by the cycles within the LRE window, in which the predictive fluorescence readings, referred to as PFC, are compared to the actual fluorescence readings, with the difference expressed as a percentage. This illustrates the high level of precision that can be achieved with LRE modeling, generating an average difference of only 0.17% over a 17-fold increase in reaction fluorescence. When applied to the entire profile, it is evident that this profile shows a close correlation to the predictive fluorescence readings, even within the plateau phase. It should be noted, however, that while this is typical for high-quality profiles, loss of conformity, particularly in the plateau phase, has often been found to be correlated with poorly performing assays. Finally, profile position can be calculated using another LRE derivative, which is defined by the fractional cycle, called C1/2, at which reaction fluorescence reaches half of Fmax. To summarize, LRE is founded upon a linear coupling between amplicon quantity and loss in amplification efficiency. This allows linear regression analysis to be used to determine values for delta E from the slope and the maximal amplification efficiency from the y-intercept. A derivative of the Boltzmann sigmoid function is then used to convert fluorescence readings into target quantity, expressed in fluorescence units. This generates 4 to 8 determinations from each amplification profile, from which an average F0 is calculated. The next step is to convert the average F0 into the number of target molecules, which is the subject of the next presentation. This completes the LRE analysis section of the overview. As always, thank you for your interest.